Welcome to Easy Microbiology.
Good morning, everyone. We are here today to present on Actinomyces israelii. This bacterial species is an important inhabitant of the human oral cavity, and as we will see, this microorganism has a variety of characteristics and properties that make it very interesting for us to explore. Let's begin our journey and learn more about Actinomyces israelii. A common inhabitant of the human oral cavity, Actinomyces israelii is an anaerobic gram-positive bacteria belonging to the Actinomyces genus. It can cause various infectious diseases and, therefore, it is important to understand this bacterium and its potential implications, especially in the presence of weakened immune systems. A bacterium found in the human body, Actinomyces israelii generally resides in the oral cavity, gastrointestinal and genitourinary tracts, and skin. An important part of the human body in its healthy functioning, it coexists peacefully with other bacteria, and only becomes a pathogen if present in large quantities. A bacterium named Actinomyces israelii is an anaerobic, filamentous bacteria that exists in different habitats. Present in the environment such as soil, water, and in the human body, the bacteria displays long and thin filaments that can grow up to several centimeters in length. According to microscopic observation of the filaments, they appear like branching chains of rod-shaped cells. Actinomyces israelii is a slow-growing anaerobic bacterium capable of forming colonies on anaerobic blood agar. These colonies typically appear white, opaque, and non-hemolytic. To survive, this species of bacteria needs specific conditions and holds great significance in terms of understanding. Actinomyces israelii is a facultative anaerobe with distinct biochemical characteristics, such as the ability to ferment carbohydrates to produce lactic acid, and unlike other organisms, it cannot produce the enzyme catalase. These features make it necessary to treat it differently when it comes to biochemical analysis and metabolic processes. Actinomyces israelii is a species of bacteria commonly found in the human body, mostly in the oral cavity, gastrointestinal tract, genitourinary tract, and skin. It is a commensal organism, which means it does not cause any harm to its host and lives in harmony with it. Therefore, it is an important considerator in both medical and dental treatment. Actinomyces israelii are an important pathogen capable of causing a variety of infections. Virulence factors such as adhesins, collagen ases, and hyaluronidases enable the bacteria to attach to and invade host tissues, making it a particularly dangerous microorganism. Awareness of the risks posed by Actinomyces israelii and measures to reduce them are therefore essential. A. Israelii has the ability to form biofilms which offer a layer of protection against antibiotics like penicillin, allowing them to survive in environments that they would normally not be able to. This is one of the many mechanisms of antibiotic resistance seen in A. israelii, and further research may shed more light into how this process works. Actinomyces israelii is a rare bacterium which can cause infection and is often associated with medical or dental procedures such as tooth extractions or root canal treatments. Despite its benign nature initially, if left untreated, this infection can become life-threatening. Hence, it is important to take prompt and appropriate action when any symptoms arise. Actinomyces israelii can cause infections when it gains entry into the body, typically through an injury or wound on the skin or mucosal membranes. These infections, if left untreated, can escalate into more severe problems like abscesses or granulomas, which can become long-term. It is thus essential to be mindful of this and take precautionary measures to avoid potential infections. Actinomyces israelii infections are caused by a bacterium which typically dwells in the mouth and respiratory tract. However, in rare cases when it spreads beyond this area, the body can trigger an immune response through the production of antibodies and T cells. This can help to clear the infection and minimize the chance of further complications. A combination of culture, histopathology, and radiographic imaging is necessary for accurate diagnosis of Actinomyces israelii infections, which can often go undiagnosed due to the slow growth of the bacterium and the lack of clear symptoms. Early diagnosis and treatment is essential for achieving positive patient outcomes. Oral hygiene is essential in preventing Actinomyces israelii infections. Brushing and flossing after meals can help remove food particles and bacteria that can lead to infection. Adhering to infection control measures during dental procedures can help reduce the risk of infection. These measures can include sterilization techniques and the use of personal protective equipment. A combination of antibiotics and surgery is generally required to treat Actinomyces israelii infections. Antibiotics are to be consumed for a number of months to eradicate the infection, and any affected tissue may need to be surgically eliminated in order to clear away any abscesses. This method guarantees fast and effective treatment. Without proper diagnosis and treatment, Actinomyces israelii strains can cause serious infections when they gain access to other parts of the body. These strains are a common part of the human oral microbiota. We sincerely appreciate your attention to this presentation.